Welcome back to Chapter 7.1. This will be the second and final part to this particular section, which covers chromosomes and phenotype. In Part 1 of our discussion, we reviewed some of our terms from Chapter 6, and we saw how some of those contributed within a more complex setting of traits, such as disorders caused by recessive alleles. Once again, in which case, in order to have this particular disorder, you would need 2 copies of the recessive allele. Heterozygous would not have the disorder, but they could be carriers, so they could have children with it. And homozygous dominant did not have the disorder either. Once again, they were fine, homozygous and heterozygous, because they had at least one dominant allele, meaning that the recessive allele that they had if they were heterozygous was not effective. We use an example of cystic fibrosis to see how heterozygous parents or carriers could have a child with the disorder. And then we finished up 7.1 by looking at disorders caused by dominant alleles, discussing that these are much more rare in the population, because they tend to kill people at a relatively young age, typically ages 3 or 4, meaning that those individuals really don't have much opportunity to have children. One exempt disorder would be Huntington's, because this doesn't affect an individual until they're much later in their life cycle, so they probably would have had children by the time they're in their 30s. So moving on, our second main idea for 7.1 is that males and females can differ in sex-linked traits. In the first part, we were looking at autosomal disorders, but now we're going to be looking at sex-linked items. So let's go back to Mendel. Mendel figured out a lot about heredity. A lot of the stuff that we know came from Mendel, including dominant and recessive. But Mendel didn't know that the genes were found on chromosomes. So as it turns out, just coincidentally, the traits that Mendel was looking at, height of the pea plants, the color of the peas, that sort of business, those traits were only found on the autosomes. So they had really nothing to do with gender. So now today we know about sex chromosomes, and we know that the expression of genes found on these chromosomes is different from the expression of genes on autosomes. So let's take a look at our sex chromosomes here. We see an X and a Y, meaning that this is a male, and we can see that the X chromosome is incredibly larger than the Y chromosome. So how does this influence sex-linked traits? Well, these 2 chromosomes they're different. They are absolutely different. And if we look up here, we see this dark band right there, indicating that there's a gene there. Where is that over here? Well, it doesn't exist because it's not on the Y chromosome. Similarly, we have a light band right here. Where is that? Well, it's not necessarily anywhere over here because there are different chromosomes. Looking at the Y, we see that there's this dark band here. Well, where is that over here? Well, it might be hard to tell, but that gene right there, it's not anywhere over here. So these 2 chromosomes are entirely different, which does factor into traits that we see in males and females specifically. So we're going to look at a few of those coming up here. So sex-linked genes, right here is a Punnett square. We can see that we have a female and we have a male, and that in order to for a couple to have a female child, the one X chromosome is guaranteed coming from mom, but the second one has to be this one from dad. It's the only way we can get that. One X chromosome from mom, one from dad. Meaning that all males, they'll only have one copy of an X chromosome because their second sex chromosome is a Y. So we're going to be looking at sex-linked genes, and sex-linked genes are those genes that are found on these sex chromosomes. And we're going to be looking at ones that are tied specifically to the X chromosomes. We're not going to be talking about genes tied to the Y. So let's consider this. If a gene is found on the X chromosome. So genes found on the X chromosomes. A female who has 2 copies of that X chromosome could be homozygous dominant, big R, big R. So on this X chromosome, maybe there's a copy of a big R, and on this one, big R as well. That female could also be heterozygous, big R, little r. So maybe the big R is on this one, the little r is on this one. Or she could be homozygous recessive, little r, little r. So maybe on this one, maybe it's little r, maybe this one it's also little r. Females will have 2 copies of that gene because they have 2 copies of the X chromosome. And once again, we're talking about genes found only on the X chromosome, genes that are not present on the Y chromosome whatsoever. How does that compare to males? Well, keep in mind males only have 1 copy of that X chromosome. So, a male could on this X chromosome have the dominant allele, R, or they could have the recessive allele, little r. Notice that the genotype for a male, for any trait found on the X chromosome, is only going to have 1 letter, and it's going to be dominant or recessive. In this case, males, our genotype is simply put R. In this case, our genotype is simply put little r. A male cannot be heterozygous for genes found on the X chromosome. They can't be heterozygous because they only have 1 copy of that X chromosome. So how does this factor in for problems with genes and traits that are sex-linked? Well, let's talk about recessive disorders found on the X chromosome. And we talked about carriers previously. In order for a female to have a problem with a gene found on the X chromosome that's recessive, she would need to be homozygous recessive. If she had that combination of alleles, this female would be affected. However, this same female, if she had just 1 big R, she would not have the disorder, whatever disorder we happen to be talking about. And obviously the homozygous dominant female with big R, she would not have the disorder either because she has a dominant allele, and you would need 2 little r's for the recessive disorder to exist. Males, it's different. Males, we are either R or little r, and if we are big R, we do not have the disorder. If we are little r, we have the disorder. So, essentially, a female can have a little r. A female can have 1 little r, and she's fine, if the second one is big R. On the other hand, males, if we have 1 little r, we have the disorder. So traits and genes and disorders found on the X chromosome, they affect males differently than how they affect females. Females, if they have this disorder, this problematic allele, they have a second allele to hopefully be there to mask it. Males, we are either having the disorder or we do not have the disorder. So notice that we only write 1 allele for males because we only have 1 allele for that particular gene. Last thing we need to talk about, once again, still same main idea, males and females can differ in sex-linked traits. You may have seen these type of cats before. They're called calico cats. And we're going to be talking about how these patterns arise within the fur color. Brown or orange, black. Like, how come it's not a solid color? Well, this is due to a thing called X chromosome inactivation. And if you know what the word inactivate means, it's pretty straightforward. An X chromosome is inactivated. So this only happens in female mammals. So any female mammal, this happens. Females have 2 X chromosomes. We just spoke about that. Males just have 1. One of those X chromosomes is fully functional, and all of the genes on that X chromosome are expressed. The second one, however, the second X chromosome, it's turned off. It's not needed, so it just kind of shrivels up and goes away. Males, on the other hand, we just have 1 X chromosome, So if the X chromosome happens to be for orange fur color, then we have orange fur in cats anyways. And conversely, if the X chromosome happens to be for the color black, because fur color is found on these X chromosomes, then that male cat will have a solid black coat. Females, it's a little bit different. Notice right here we're looking at homozygous black. Here we're looking at homozygous orange. And in this case, one of these X chromosomes is going to shrivel up because it's inactivated, and the other one remains present and it is expressed, meaning that because this female, all of her cells are going to have the same allele, regardless of which of these chromosomes is inactivated, will have a solid color. Same with this one. All of the cells in this cat's body are going to have either this X chromosome or that X chromosome working. The other one gets shut off. So it doesn't matter, this female cat's going to be solid in color, orange. However, looking at this one, we're looking at heterozygous cats, and this is where we get this cool pattern. We have two alleles, one for the orange, one for the black. Randomly, one chromosome is going to be shut off. So in this case, we see the orange one shut off, and the black one's active. And in this case, we see the black one shut off, and the orange one is active. So this cat could have some cells with black, like right here, and in the very same living thing could have some cells with orange activated, meaning we'll have spots of orange. Now, this is different across all cells of the body. The cells up here, obviously, with the orange pattern, they have this particular combination working, meaning that the black is shut off. But over here, the black is turned on and the orange is shut off. So each cell in the body is a patch, and they are different, resulting in this pattern color. Male cats, at least of the calico version, they can only be solid in color because you can't turn off an X chromosome if there's only one copy. You can turn off an X chromosome if there are two copies of it, though. So this concludes section 7.1. Make sure you have all of those notes filled in, and make sure that you ask me any questions if you haven't done so already while you were working on this.